for the opportunity to present at Grand Rounds, Dr. Ellenbogen, and to Dr. Code, Dr. Ojman, Dr. Hellman for helping to craft this training year, which has been off to a great start. And I'm really enjoying it. And I've learned extensively clinically and getting going on research. In terms of what I'll talk about today, this was really probably the project that I hold most dear from my residency research. It's in the area of cognitive neuroscience, which had always interested me as I was considering a career in neurosurgery. Specifically, how can we leverage neurosurgery to ask interesting questions about the human brain and what makes us human, and our ability to perform very complex calculations, and how can we probe the brain to answer these types of questions. So the title of my talk in the paper is Semantic Encoding During Language Comprehension in Humans at Single Cell Resolution. So this is a study of prefrontal single unit activity in the context of language comprehension. And I think neurosurgeons in particular, we're not only able to contribute to this field of human cognitive neuroscience, but I think we are uniquely able to. The field is dominated by functional imaging studies, evoked potential studies, which are incredibly valuable, but that's 99.9% of the literature, and I think we can contribute through intracranial recordings, single unit analysis to really understand these complex behaviors. So I have no financial conflicts of interest to disclose. I just first wanted to start out by commenting on the team that's behind this work. I worked with Dr. Williams, who's a neurosurgeon, neuroscientist at Mass General. Clinically, he is a specialist in functional neurosurgery, both movement disorders and epilepsy, and was a great mentor and is a fantastic scientist, very creative mind, and really has a passion in the lab. Worked with several postdocs, Mohsen, Jill, Ray, all pictured here, and in the bottom middle is Arjun Khanna, who's a co-resident of mine. And we had a superstar collaborator at MIT, Evelina Fedorenko, who's an emerging world leader in neurolinguistics. And as I learned, linguistics as a field is very territorial, very passionate people, and it's good to have a very good ally when making contributions and doing research in the field. I just wanted to give a summary of some of the other projects that I focused on in this realm of cognitive neuroscience. The first two have been published, and the one that I'll talk about today has been accepted. We asked questions about single unit responses in the setting of judgment, specifically in tasks in which there was no clear choice of right or wrong, but where there were various grades of ambiguity. So for example, these were all done in the setting of awake DBS surgery, asking patients to generate an opinion as to whether or not a certain image was safe, like a person crossing a street in varying degrees of proximity to a car, and found that there were neurons that modulated and were reflective of not just the choice, but the actual degree of ambiguity of the situation. Another major paper that we put out was in the area of theory of mind, which is a popular buzzword in cognitive neuroscience now. And it refers to our ability to quickly and rapidly and continuously update representations of other people's thoughts that we have. And so it's not just our world models don't only consist of what we see directly, but hypotheses that we generate about other people and what they're thinking, and allows us to anticipate their actions. And in medial prefrontal area, we were able to show that there are neurons that selectively respond to content in listened passages that were reflective of inferences that other people were making. And we published that. And as I mentioned, the paper that I'll talk about today relates to single unit responses in the setting of language construction in meetings. So here I'm talking about our ability to rapidly listen to a sentence or even just a few words in an utterance and create a very rich picture or meaning construction of what that intended speech or linguistic input was intended to communicate. So brief outline. I'll spend a good amount of time at the beginning talking about models of speech and language and how they're evolving in the past decade. I think many of us are familiar through our intraoperative awake mapping experience of the classic model, and I just want to point out some of the evolving areas in the field. I'll talk specifically about semantic processing and how it relates to prefrontal areas. And the reason I have this image here to the left showing this low-grade lesion basically taking up the whole left frontal pole is, I think as surgeons listen to this talk, they quickly say, well, you're measuring neurons from a non-eloquent area that I've taken out tens, if not hundreds of times, and patients wake up fine, maybe with transient aphasia, but move on to communicate effectively without any appreciated deficit. And I hope by the end of the talk, I've provided data to support that these areas are important. They're not essential in terms of they are broadly distributed, but that we can continue to be very aggressive with tumor resections, but appreciate the fact that these frontal areas are involved in language processing. And then at the end, I'll also talk about a new area in studying not semantics or the meaning of language, but the syntax or the rules by which we construct clauses, sentences, and these rules are necessary to actually to arrive at conclusions from linguistic input. So there's as we all know, especially at this institution, very rich history of neurosurgeons involved in speech mapping. Obviously, the contributions from Penfield are well known, as are from Dr. George Ogeman, Dr. Jeff Ogeman, where through intraoperative direct electrical stimulation, we're able to not only identify areas that are essential in generating naming or other speech tasks, but actually map out the probability distribution in the perisylvian cortex of where these areas are. And then as Dr. Sinai, Mirzadeh, and Berger published in 2008 in the New England Journal, able to correlate how this method of intraoperative speech mapping actually leads to better outcomes in patients longer term. And as we look at the literature in functional imaging, evoked response potentials that has come out recently, a trend that is noted is that when we move from tasks such as naming to actually understanding or creating models of generating complex meaning from complicated sentences, what we begin to do is move beyond the perisylvian cortex and begin to invoke frontal areas as well. So language is very rich. It's not simply naming, as we know. So if you take this linguistic input on squirrel, the, fall, a, made, nest, tree, the, every, it's nonsensical. Your brain initially tries to parse it together in a meaningful way, but very clearly, if we were to reorder things into a syntactically sensical message, you can very rapidly, within the order of hundreds of milliseconds, create a very rich understanding of how this linguistic input was intended to be communicated. So it's this interplay between individual meanings of words and how they are linked together that I refer to when I use the word language. So this is really, if you just think about the physics here of transforming a air pressure wave that's generated by someone's voice when they say the child bent down to smell the rose, and the complexity of the spectral data within that waveform and how that gets transformed into a complex meaning within, again, a matter of seconds is quite remarkable. So first there's that construction of the phonetics. So taking that frequency data and pairing it to known phonemes that can be used to construct syllables, and then syllables obviously to construct words. And in the linguistics, field of linguistics, lexical refers to the actual form of words, either in how they are heard, the auditory form, or in the orthographic form, which is how they are written. And then from there, we can start to build upon the semantics, actually attaching meanings that can be context dependent to each of the individual words. And and then using syntactical parsers to link these together, again, adding another rich element to the meaning construction. And finally, we end at the creation of a proposition, or what was actually intended. And the field has begun to use the word unification as the term that basically unites all these features. And also that term includes non-linguistic cues such as social cues or environmental cues that can sway how one person interprets a linguistic input. So the classical model of auditory language begins in the cochlea. Obviously, the inner hair cells there are the first pass of transforming the air pressure wave into an electrical signal by decoding along a frequency spectrum. And then that information through the cochlear nerve is brought in through spiral ganglion to the cochlear nucleus, and very quickly within the lower brainstem has a bilateral representation as it decussates or crosses through the trapezoid body to the superior olive, and then ascends in the lateral lemniscus to the inferior colliculus, and then to the thalamus. And so this type of calculation in the lower brainstem is probably most helpful in reflex reactions in terms of calculating time lag from left and right and determining where exactly in horizontal and vertical space the sound is coming from, but clearly is not at the point where we're starting to construct meaning from an intended speech. And it's important to recognize that this input, it ends up in both of the Heschel gyri bilaterally in both of the superior temporal lobes, where the spectral and phonetic data is decomposed. There's thought that the left initially starts to do that more in a linguistic sense, and the right more in a pure auditory sense. But it is important to realize that this is bilateral. And then Norman Geschwin, who's probably one of the most highly contributory behavioral neurologists of the 20th century, actually was able to go back and look at Wernicke's and Broca's work and integrate that into the Wernicke-Geschwin model as it's referred to today, and what we use in any surgery as a workhorse model, where Broca's area is connected to Wernicke's area through the arcuate fasciculus. But even Geschwin, in some of his writing, basically shows that model is not sufficient for explaining how we really can do complex comprehension. So here, I think it's best shown in a simple case of a 22-year-old woman that he published that had ischemic injury. I believe this was from carbon monoxide poisoning. And the ischemia, as you can see in the post-mortem slides here, really involved inferior temporal areas. More anteriorly, we have dorsal, frontal, prefrontal areas as well. But importantly, what's preserved here is basically that workhorse area for speech, the inferior frontal gyrus, superior temporal gyrus, arcuate fasciculus. So this patient was able to do basic things like repetition with the intact arcuate, but when it came to clear evidence of verbal comprehension, complex command following, was not able to do that. Could repeat or fill in previously learned phrases such as roses are red, would then fill in violets are blue, and could even learn new songs in a phonetic or an auditory sense. But there was no sense that this patient was actually able to generate propositional or meaningful new speech. And he writes that the patient's failure to comprehend presumably resulted from the fact that the language inputs could arouse no associations elsewhere in the brain. And since information from other portions of the brain could not reach the speech areas, there was no propositional speech. So Hickok and Peppel have, this is just another more complex model of how language processing may actually occur, involving some of these areas outside of the perisylvian cortex. So they identify a ventral and dorsal stream, where the dorsal stream is really more responsible from an articulatory point of view. So whether the linguistic input is visual or auditory, there is a projection to frontal areas that then are involved in activating areas motor related that are involved in articulation, whereas the temporal pathway shown here in pink is involved in the lexical interface, so mapping those inputs to word forms and then starting to do essentially probabilistic analysis on the word combinations that then begin to allow us to put meaning onto those word combinations. But notably here in gray, they kind of say the conceptual network is widely distributed, which some people may interpret as just unknown. And then this is kind of an area that I think is interesting and where a lot of work can be dedicated to further understanding. So this was a paper that was coming out just as I was preparing for my research time in residency from a Gallant group at UC Berkeley that had patients listen to podcasts of NPR while they're in the MRI scanner. And then each word, they replaced each word with what's called a word embedding, which is a high dimensional vector where the entries of that vector correspond to word similarity. And I'll talk a little bit more about word embeddings later because we use it in our work too. And then it allows you to replace a word with an actual quantitative vector representation and then do complex modeling. And so here, every voxel was modeled as a function of those word embeddings. And if it shows up in yellow or any color here, it was a significantly correlated voxel. And then the heat of that voxel on the maps here correlate to the p-value of that association. So we do see bilateral and broadly distributed areas of activation that seem to correlate with semantic content of the input that the patients were listening to. They then also did an interesting analysis where they took the model fits and performed principal component analysis of those model fits and reduced that huge space where each voxel is represented to a smaller dimensional, 4-dimensional space, essentially, where they could then project the word embeddings of each word back into that PC space and see how those words organized along those principal components. And you can see, if you look at the top left here, like principal component 1, to one extreme tended to be locational and the other social, emotional. Obviously, these are not like perfectly delineated, obvious word clusterings, but it showed that perhaps there is, in the setting of a broadly distributed semantic system, some areas of local organization. And then this is a model that I've kind of adapted from this book that I think is a compelling one. And I've kind of come up with the term cycle model here. But it basically says, it offers a role for the frontal cortex in integrating linguistic meaning to create these concepts or perform high-level comprehension, where whether the input is visual or auditory, visual from occipital, auditory from superior temporal, that initially within superior temporal sulcus, there is that decoding into the, what we call the type of word. So, you know, if you hear the word flat, you might initially think of the shape of an object that's flat, but that orthographic type has many different meanings depending upon the relative context. So flat tire, flat beer, flat note. Each instance of that flat is, we call a token, and that token can take on various meanings depending upon the context. And we have projections from the temporal area, the uncinate, the inferior frontal occipital, the arcuate. Left out here, there's also parietal frontal projections through the superior longitudinal fasciculus that continuously feed this information. And again, this is theorized to transform what are types of words to actually token integration of complex contextual meaning. So we worked with, as I mentioned, Ed Fedorenko from MIT and started to look at some of her data. And what she was doing is having participants listen to different linguistic input in fMRI and then compare contrasts of that input, depending upon whether it was sensical, nonsensical, or structured. So things like non-words here showed in the bottom, the nephidas mas grande glissi, absolutely nonsensical, just sounds like a list of words, versus Jabberwocky, which the flompate smatagisk on the end sounds like a sentence because there are functional words there, but again, you cannot generate anything meaningful from that. And then word lists, which are just lists of words that are not ordered in a sentence, and then sentences themselves, and showed that as you go from non-words to sentences, that you tend to see frontal activation or broader activation for all intents and purposes. And that was interesting to us, specifically because we often access the frontal lobe in surgery, and given that we call it non-eloquent cortex. If you actually look at regions of interest, the left middle frontal gyrus in her studies had some of the strongest differences between sentence-related activation and non-word activation. Again, this is a broad effect, but the left middle frontal gyrus, often an entry point in deep brain stimulation, was seen. And so when we took an even finer look at some of that data that went into that bar graph and compared left middle frontal gyral activation in response to sentences versus lists, we saw a highly significant difference here. And then if you go from a bar graph where everyone's grouped to actually looking at individuals, each individual is a blue dot here, each one on their own had a higher bold signal activation in the middle frontal gyrus in response to sentences compared to word lists. So we hypothesized that the prefrontal cortex aids in language perception or comprehension in a context-dependent integration of the semantic input. And so we set out to test whether or not we could isolate single neurons in that area and to test for this association of a context-dependent role for these neurons in linguistic processing. So we adapted our standard deep brain stimulation microelectrode recording. During my residency, we almost did all deep brain stimulation awake with microelectrode recording. And we would use the 5 tungsten microelectrodes through a middle frontal gyrus entry point, the same entry point that we would use for the placement of the DBS electrode, but paused in the cortical layers and used a lot of TCL to kind of dampen the movement with pulsations of the brain. And we had some success in actually isolating units, so putative units in the operating room that we then sorted offline post-operatively based upon waveforms. And so what we would have the patients do is listen to various linguistic inputs while they're awake, a combination of sentences and word lists. And then post-op, we were able to align our isolated single units to the sentences as shown in this figure here. So then the question is, how do you analyze a data set like this? We have a bunch of words, very sparse data. The average neuron here is not firing particularly fast, but we wanted to do per word analysis because that's really where we get both interesting questions and then statistical strength. So going back, we leveraged that word embedding technique that the Gallant group had used in the paper I mentioned earlier. And just to comment a little bit on how these are generated. So it's something that's evolved out of the natural language processing world, where folks at Google were aiming to come up with vector representations of words. And what they would do is use a very simple neural network where you'd go through a corpus, say every article published on New York Times or Wall Street Journal, and you'd feed in each word and then train a matrix to predict the probabilities of what of the adjacent words in the sentence. And they weren't really interested in creating a model that was highly predictive. What they're interested in is, as you train this, how does this matrix evolve in the middle such that you can take out a single column which corresponds to a single word and actually use that as a vector representation of the word. And so when they looked at how these vectors and words were aligning in this high dimensional space, you can choose any dimension, but here we have 300. the words tend to cluster based upon meaning when you just look at it with the human eye. And then even more interestingly is that they're actually additive properties of these vectors, where if you, for example, if you took the word embedding for the word king and you subtracted the vector boy, but then added girl, the resultant vector in that space would be closest to the vector for queen. So this was a successful attempt to actually come up with vector representations of words. So we used that, and we did k-means clustering of the words in our vocabulary. And we did this several times with different randomizations of the seeds for the k-means to figure out, how many groups should we use, what's reasonable here, and tried to remain as agnostic to what the words were themselves as possible. It's to generate these word clusters. And what we found is that there were a fairly good thematic labeling opportunities for these words. So things that were branching out semantic, in terms of semantic themes such as food, animals, family, names, actions tend to cluster, locations or descriptive words tend to cluster, or even words about or words of environment themselves. So we chose 9, and our basis for choosing 9 was, as we repeated that clustering analysis, we could simulate this as many times as we want, but after doing it several hundred times and using what's called the silhouette metric, where it basically asks how close are what is the greatest difference between words within a cluster compared to words in other clusters, and then optimizing that metric as a degree of cluster separability in your cluster model. The silhouette metric was greatest for 9 clusters when we repeated the simulation. So that was our basis for choosing 9. And so then after having these 9 clusters, we went back and looked at each individual neuron and compared firing rate activity in response to words belonging to that group versus words outside of that group. And what began to emerge, we had isolated a total of 220 neurons across 10 patients. In a subset of them, 47, we could identify at least 1, at times 2 or 3 groups where there was differential activity of that neuron. So here, 2 examples here where the neuron for the group of interest, for here, this neuron had an increased firing rate 100 milliseconds to 500 milliseconds after it heard words that were food related or descriptive related. And then here next to it is one that tended to respond to family and relationships. So we found this pretty remarkable, and used other metrics to characterize how selective these neurons were. Here is a selectivity index where we took the difference of the firing rate in response to the preferred group to responses to other groups, and then over the sum of those 2 values. So 1 would be the most highly selective and 0 would be the least. And the 2 colored in ones here are the neurons that I showed on the previous slide, just to demonstrate that they're not drastically different. And then what we did is to say, well, how specific is our response to the word vocabulary in our study structure specifically? And so we subsampled 60% and showed that our findings was robust, even if we did random subselection of 60% of the words in our study. This is a heat map showing the heat represents the number of neurons selective for that particular word domain. So most of the heat is along the diagonal, suggesting that these words these neurons tended to be selected for 1 and sometimes 2 neurons. There's very little cross selectivity. And this is the histogram of the number of neurons significant for a certain number of domains. So 2/3 of them were selected for only one domain. And then, so as I mentioned earlier, we had the ability to compare this effect across different degrees of linguistic input. And again, we're measuring here in the frontal lobe. So if we go back and look at these neurons in response to the same words, but now in a word list rather than a structured sentence, these effects tended to fall out. So we had many fewer neurons that met statistical significance. And this is the same heat map showed where yellow is zero selective neurons. So most of this map, there are no selective neurons for these words in the setting of a non-ordered word list. So here we're starting to conclude that these responses were context dependent, and thus fitting with that model that I had mentioned earlier. This gives a selectivity index for these neurons in response to sentences versus word lists, and then the same data shown in a histogram showing that the selectivity index for sentences tends to be shifted closer to 1 than for word lists again. So again, this is consistent with the previous conclusion. If we reorder our words and word groupings in a hierarchical structure, we can then say, we can then talk about adjacent word groups. So where food and animals tend to be closer than food and environment for whatever reason. And then we plot the activity of the neurons in response to either the cluster of interest, the domain of interest in group 1, or the most adjacent, group 2, the next most adjacent, group 3. In the setting of sentences, we see a very high preference or differential activity in response to the preferred domain. And again, in the setting of word lists, this tends to fall out. We did population level decoding as well, modeling and decoding, where we grouped all neurons together into an ensemble-based analysis and did a categorical logistic regression model, fairly simple, but trained it on 70% of our data and then tried to predict the category of a word in the remaining 30%, and found that, as expected, that we could decode with more neurons, and there tended to be a limit to that performance increase depending upon the word cluster. Part of this is a statistical phenomenon. It's harder to decode with the greater number of word clusters that you're trying to predict. These graphs are normalized to the increase above the baseline performance expectation. So that would be just guessing the correct category by chance. But we did see that around our selected cluster of 9 neurons, we see a plateau in this increase. So like any study, your conclusions are only as good as your controls. So we set out to explore different opportunities to test how robust our findings were. One of them was the use of homophones, so words that had the same sound but different meaning. Shown here, their activity change shown in red. So the activity change here means the difference in firing response in a neuron in response to, for example, the word red versus red in the setting of reading, and then compare that to non-homophones, so words that belong to the same semantic category from our clustering, but were not homophones. And the differential activity, despite having the same phonetic sound, was actually greater in those same neurons in response to homophones than if you compared it, the differences of the words within their same group, suggestive that, again, these neurons were more responsive to the actual linguistic meaning rather than the phonetics of the words themselves. Prefrontal areas are obviously involved in working memory, so it's important to control for that. Proxy for working memory can just be activity in general. So you could hypothesize that our effects here in terms of more effects being seen in sentences versus word lists is purely a working memory phenomenon. But we actually see higher activation in neurons in response to word lists than sentences themselves, which suggests, so that counters that concern. Additionally, if you just plot all of our neurons in terms of their activity along responses to word lists versus responses to sentences, and then highlight the neurons that here in blue just had differential activity between sentences and word lists, and those that were significant for semantic selective activity, we see a non-overlapping distribution of neurons in that case. So again, suggesting that our semantic selective data was not generated by a pure differential effect in firing rate between the 2 trial types. We also wanted to create a way to do analysis that was independent of the clustering model ourselves. So we, similar to that Gallant paper, we performed analysis where we modeled all the neuronal activity as a function of the word embedding, and then extracted the weights for that linear regression fit, and then concatenated the weights across all the neurons, and then performed principal component analysis to create this transformed distance space that we could use and analyze. So if we look at the distance between words in the pure word embedding space, so before that logistic regression and the one after it, we do see a high correlation. Now, if you take any set of vectors and do some sort of correlation and transform it, you're going to see a correlation, but the question here is that just, is it statistically significant or not? And so what we did is we actually randomized the pairing of the word and the embedding and repeated this process. And here we showed the correlation values that we generated through that random process in blue, and our actual correlation is an extreme outlier, suggesting that there actually was some meaning in this regression and transformation. This essentially shows the same effect, but binning the data in terms of distance quantiles rather than in continuous axis. So conclusions from our study here include that prefrontal neurons do appear to be active in linguistic processing, and they tend to demonstrate activity that is context dependent. And then this, provides some supporting evidence for the cycle model or other models that I mentioned earlier in the talk. It is, one thing to be able to go into the middle frontal gyrus and decode across many different semantic domains. And so looking at one little area is consistent, we would say, we're not suggesting that this area does it all by any means. We know that that's not true from our surgical experience at the very least, but it does show that this information is likely broadly and redundantly represented across cortical areas that are involved in these constructions of semantic representation. So, there's good data, interesting experiments to confirm that cognitive information in general has a bilateral representation. This is a mouse study that was published in Nature in 2016, where a mouse was trained to learn the position of a pole in its environment, and then after some delay had to perform a whisker lick in one direction to demonstrate where that pole was. And so at various trials, the mouse received small degree of unilateral inhibitory stimulation or a large degree of bilateral frontal inhibitory stimulation. And also they did this with and without a corpus callosotomy performed. And so if a non-corpus callosotomy mouse did this and had small degree of inhibitory stim on one side, was able to reconstruct the correct answer at near baseline, suggesting that even though there was a left-sided inhibitory stim, that the information was stored elsewhere and could be reconstituted to generate the correct response. But if the corpus callosum was cut, that performance decreases. And then in either case, with an intact or a non-intact corpus callosum, if there's bifrontal inhibitory stim, the ability to correctly respond is diminished. So again, suggesting bilateral and broad distribution of cognitive information in a simple but elegant study. In humans, fMRI data has showed similar findings. So here, participants were exposed to different degrees of linguistic input. So a backward story, so literally just read in backwards, so you couldn't make any sense of it. And then non-ordered wordless sentences or paragraphs. And with each increase in the complexity or the meaningfulness of the input, we see bilateral activation and the involvement of frontal activation when we start introducing sentences. So I think this has a rich future in terms of next steps to take this type of study and what we learned from it. One is questions about syntax. So syntax has been an aspect of field of linguistics of interest and hypothesis, and a lot of work for, for centuries. And Noam Chomsky is probably best known for proposing the universal grammar theory, where all humans are innately born with some degree of a grammatical structure. This has come under debate as to how separable semantics and syntax really are. It seems that probably Broca's area is definitely involved in syntactical organization, but it would be interesting to explore this type of a question. Also, there are theories as to what is the dynamic by which we create these meaning constructs in the setting of language. How do we recruit memory? Are we generating potential meanings in parallel versus series? There seems to be some debate as to whether, or some likelihood that semantic generation is a parallel process, whereas syntactic processing tends to be in series, where you commit to one potential way to interpret a sentence, and then when it doesn't make sense, you go back and you consider another one. But when you hear even a single syllable, you're already generating multiple possible candidates for what that word actually is and what its meaning is in parallel. Studying non-linguistic inputs such as the environment, social cues, etc., and how that feeds into this unification idea. And then modality and variance. What I presented here is purely from auditory input, but it would be interesting to explore differences as to whether something was read, or if in bilingual patients, is there a difference between language response in one versus a different language. And then, a great limitation to our study here is the fact that we're looking at one area in isolation. And I do think that some of the modalities here that we're putting in place in epilepsy patients, Dr. Ojman, Dr. Ko, Dr. Hauptman, using SEEG and Benke-Fried electrodes could allow us to expand the scope of how we are looking at language from my current study here to considering broad areas and how they interact in this complex process. Also allow us to do, quite frankly, just collect more data, because we'd be outside the operating room, where it's a little bit easier to do that. Just going to make a couple comments on the semantic versus syntax question. So the evoked response literature, which is basically studies from EEG, so capturing evoked responses at the level of the scalp, show that you can detect like semantic errors versus syntactic errors at the level of the scalp. So the N400 is a deflection that tends to be central on the EEG when someone hears a very unexpected word given the prior context. So an example is the paratrooper jumped out of an airplane. That would be highly expected, but if you replaced airplane with ostrich, that would be highly unexpected, and you can start to detect these types of errors. There's also one that tends to be more specific for syntactical errors. So the plurality error in this sentence, the paratrooper jumped out of an airplane. But if it was jumped out of an airplanes, that's not really an error in meaning so much as it's an error in grammar or syntax. And there's a left anterior, tends to be a unilateral negative deflection that is seen in those contexts. So if you can see them at the scalp, you can probably see them intracranially, and exploring that I think would be of great interest. We started to do that to some degree in a study that I was a part of during my senior residency, where we actually wanted to do analysis on a per word basis, and you can use what's called constituency parsing, where each word in a sentence is labeled based upon its ability to be parsed into a more cohesive unit of meaning. So if you look at this sentence, the ambassador can then be grouped into the noun phrase, and then who negotiated the deal. At deal, you have another point of integration where this clause, this subjunctive clause describing the ambassador, can be integrated, and then you can link it all at the end, was a skilled diplomat, when you have the verb phrase. So at each point, you can study how the linguistic information is integrated, and use these time points as effectively as trials in an analysis of intracranial data. And there is one group that has studied, or probably more groups at this point, but one group that put out a nice paper in 2017 that looked at each of these junctions in patients who had depth electrodes in for epilepsy monitoring, and showed that in superior temporal sulcus and also in inferior frontal areas, that the degree of high gamma power on those electrodes tended to correlate with the number of nodes that were closed at one of these moments of parsing in a sentence. So here up top, if the number of nodes that was open as it increased, the high gamma power relative to that word was greatest. Whereas here, you know, when in the inferior frontal gyrus, similarly, the number of nodes that were closed at the end of a sentence tended to correlate with the power on those contacts. We looked at simple analysis comparing subjects versus objects, so more of a grammatical comparison, and started to see differences in some neurons. Again, these are prefrontal in response to this, but this is very early work and hasn't been completely developed. But just want to finish up by thanking again the team that I worked with, which was a huge support and just great, generous people to work with, super smart and fun. Also supported by the NREF grant during that time and a T32, which helped support the research. So thanks everyone for your attention, the opportunity to present today.